name is Drol. I'm a PhD student from Dr. Patrick Schnabel lab studying on the inflorescence and the leaf architecture. It's an honor and pleasure to introduce our next speakers. Dr. Christopher Topp. Dr. Topp is an associate member and principal investigator at the Donald Danvers Plant Science Center. Chris he earned his PhD in plant biology at the University of Georgia, and did his postdoctoral works in the lab of Philip Banfi at Duke University. He's currently co-chair of the North American Plant Phenotyping Network Executive Committee and a former chair of the Phenotypes Conference. His team develops phenotyping tools and analytics to measure the growth and the environmental interactions of plants above and below ground. They use these tools to query the genetic and mechanistic basis of plant form and function. Today, he's going to talk about his team's current study using 3D imaging to monitor and dissect plant inflorescence and the root architecture. Please welcome our next speaker.
Can you all hear me now in the back just fine? Okay. Thank you so much for the opportunity to speak with you today, to the graduate students. You've done a fantastic job. This is a difficult slot, though, I got to say. Sort of the weight of history is hanging here on this stage. So I have to admit I'm a little bit, just a little bit taken aback by the incredible work of Dr. Chase. So I guess today I would like to perhaps take the audience a bit into the future, and hopefully provide some entertainment and some education along the way. Okay, so without further ado, my group is interested in root environment interactions, root growth, and ultimately how shoots and roots are integrated together. And to do so, we develop a number of imaging and analysis technologies and frameworks that allow us to answer these questions. So if you've seen one of my talks, you've more than likely seen this glowing spinning root here. This is literally we are watching carbon being pulled into the leaf through photosynthesis, converted into sugar, and allocated to the root systems in real time. About 20 minutes, a maize seedling can capture CO2 and push it down into the root. This is using a method called positron emission tomography. So these are the type of methods that we develop to really get integrated information about the plant. The reasons why, we think back about ideal characteristics or idiotypes of green revolution wheat. We know all these idiotypes quite well. This is a concept developed by C.M. Donald in the late 60s. But what you might notice here is there's quite a large gap, and the root systems were only mentioned a few times. And essentially, we know very little about the ideal characteristics of a root system for a given environment for agronomic production. Obviously, roots provide all the water and nutrients to the parts that we normally think about of a plant. Root traits are poorly understood. I think this audience knows better than most. They're incredibly difficult to breed for, and we know almost nothing about the genetic control of roots. But obviously, many of the big problems that we face societally, globally, certainly agriculturally, have a foundation in root systems. And so a real understanding touches on just a vast number of these big questions or grand challenges that we face. So we think a lot about root system architecture. This is the spatial and temporal organization of roots in the soil. And this is a classic image of prairie plants that grow around my neck of the woods in Missouri. And you can see just the incredible phenotypic diversity. Of course, this where and when a plant decides to put its roots has really important consequences for its ability to compete for water and nutrient resources, which are heterogeneous and ephemeral. So obviously, deep-rooting plants have made a bet, so to speak, that they'll be finding water, for example, in the subsoil. Everything comes at a carbon cost. And so plants, in a sense, have to make these decisions. Why don't we know more about roots? Well, these are the classic method. This is an image from Weaver's group back in 1917, basically an archaeological expedition. For those of us in this room that have ever tried to understand roots in the field, it really hasn't changed. What we can do with the samples after we get them is a different matter, and I'll tell you more about that today. Cases where we actually do understand the genetic control of a quantitative root trait, the importance is quite profound. So IR64 is a rice line widely grown in the world, known to be fairly drought sensitive. A one base pair mutation in a gene called deeper rooting 1 or Drol1, which was found in a landrace, transforms the root system. And we know a little bit about the mechanisms of how this works, but in the field, this can have a major impact in a dry scenario. It provides a huge benefit. And I think this is really the tip of the iceberg, and something that my group is especially keen to explore. So again, we think about root systems. This is again a classic image, tens or hundreds of thousands of roots, hundreds of cubic meters of soil, 3-dimension, incredibly complex structure. And yet, if we zoom into each of these individual root tips, there's a whole world going on there. This is an SEM of a root, I think a barley or wheat root. We can see the hairs, the root hairs, the rhizosheath, the pores. And so it's a major question as to how the collective behavior, so to speak, of all of these individual roots add up to the emergent property of the architecture. Very scales that we have to put together. So one of the sort of obvious ways we can do this is thinking about gravitropism, which of course happens at each individual root tip. We know quite a bit about how that works. So we work with some genetic mutants, for example, that they don't sense gravity the way they should. You can see here. I don't think I can do this on the screen. Maybe I can, but this root is growing straight up. They just sort of don't sense gravity. Now, writ large, this has a profound effect over the root system architecture. So these are the type of connections that we're trying to make in my lab. So we have a number of different phenotyping platforms, and this is one of the earliest ones and one of the simplest ones, and certainly has quite a few limitations. But it's an optical projection tomography system, which is a fancy way of saying we grow plants in a glass jar full of gel, spin them around, collect a series of images. But we can do this pretty quickly, and of course we can change them. And what it really allows us to do in the end is get an entire, complete, essentially perfect 3-dimensional representation of the root system. And this is hard to get by other means, right? It's very hard to get, and it gives us the ability to really measure a lot about the root system. Additionally, it's non-destructive, okay? And non-invasive to an extent, at least. And so we can use this to compare genetics, right? So these are two maize inbred lines. They obviously start out very similar, one primary root, but the decisions they make, the allocation to growth in various ways, to elongate an existing root or to make a set of new roots through branching, has pretty strong effects quickly on the architecture of the root system. So we've developed methods, basically computational methods, to match up each of these 3D models over time. We have an automated imaging system. We image over a week, 24/7, something it's hard to convince a graduate student to come in and do. But we were able to automate it, and essentially we can track each individual root in this root system over time using computational methods, and literally explicitly understand these relationships. And we can do this for any number of sort of attributes of the root system that we'd like. Additionally, this allows us to take a function valued approach. Sorry, that bar is in the middle, but says function valued quantification of growth patterns. And this allows us to understand really the dynamics of how the root systems are changing over time. We can see, I'm not going to belabor the different patterns we're seeing here, but we're able to take basically a complex growth process and turn it into 3 parameters, which really describe these curves and this first derivative here. And we can compare those directly. So we can now use these as traits. So we have now 3 parameter values that we can use as a trait to understand genetic or environmental influences on root growth. Furthermore, in this study, and this isn't a definitive study by any means, but in this study, we're able to see the genetic signal persisted from a seedling into a mature root system, and also, of course, from an artificial gel into the field. And so these are what we would call root crowns here. So this is essentially shovelomics. We dig these with the shovel, and I'll talk more about that in a minute. We can scan these on an X-ray system and get a three-dimensional representation of that root crown. And so now we can compare apples to apples. The three-dimensional properties of B73, Missouri 17, and their hybrid are much more similar to the in the gel and in the field to one another than and seedlings per se, or an artificial environment like a gel per se. So we can see all of the B73 genotype clustering together here, MO17 clustering together despite the age and environmental differences. The hybrid's a little bit different because, well, we get hybrid vigor, of course, and we don't see it very early on. So again, it's a useful tool. It doesn't give us all the information we want, but it does give us quite a bit of new insight. So what my lab is known for, what we do every year, is basically we grow a lot of different varieties of maize, inbreds and hybrids. We dig them up and we take these sort of formal portraits of them, right? So you can see you can see the complexity, obviously, and you can see differences by eye because humans are very good at pattern recognition. But what we have to do is turn our inherent processing into something quantitative, and we basically have to train a computer to do that. And I'll tell you a little bit more about that as we go on. So, you know, it's not pretty, but it's effective, right? So we do this by the thousands every year. We have a feed trailer, we drive around, and you know, we dig up corn. That's sort of what we do. But you'll notice here that we have this imaging system set up inside of the trailer. And this is because we obviously, it's a limited amount of information we can get if we hold the root system up to a pin board or get a ruler or a pair of calipers. Instead, we use a computer vision. So this is a computer vision pipeline called DIRT, and it was developed by a good friend and colleague at University of Georgia, my alma mater, my old department, Alex Bucksch. And it's on Cybers, so any one of us in this room can use it. We can upload our root data. You just have to sort of take this formal portrait, and you get a lot of information about root systems. And so one of the ways we're trying to tie structure to function is by working with genetic lines or populations that already have agronomic properties of interest. So we've been working with the Illinois long-term selection experiment for protein. This is a fascinating piece of maize genetics. It's a recurrent selection scheme that started in the late 1800s and has gone on for well over 100 years now, recurrently selecting for either high or low seed protein. I got interested in this as a postdoc because obviously the high protein is really good at getting nitrogen in the roots, and that's what is making the protein, right? So I assumed that there had been some indirect selection for root traits, and we would see some divergent root architectures. And in fact, this gel system, it's quite easy to see. So what we'd like to do now is to map and understand the genetics that is controlling these differences, which are plausibly related to enhanced nitrogen capture in the soil. That would be an important goal for us. So differences aren't quite as clear, but statistically, we can measure difference between these two lines all the time. So we have a mapping population between these two. It's been intermated. We call these the IPSRI. And I'll show you some GWAS results from that in a minute. But first of all, obviously capturing the roots themselves only gives us superficial information. It's high information content because it is the nexus of all the root system, but we don't really know what's happening below. At University of Illinois Urbana-Champaign, and you can see here these white things in the ground. These are 6-foot-long tubes. They're clear tubes, so they're put into the ground by this. You take a Giddings and you pull a soil core, you push these tubes in. It's called a mini rhizotron. You can then take a camera, push it down the tube, and any roots that happen to be growing along the tube, you get an image of. So we get some estimation of roots at depth. So the idea is we're combining high information content root crowns with some information at depth and trying to understand shared genetic control of these different ways of sort of incompletely measuring the entire root system. We've done this at some scale. So just to give you an example of sort of how data looks. If that's the soil, you've got the corn plant, this is depth, and this is root length density. You can see, for example, that this high protein line, which is good at getting nitrogen, has a much greater root mass, and it is a little bit deeper than the ILP line, which makes sense with what we know about nitrogen, which is of course soluble and moves through with the water column. So this has been a really nice combination for us. And again, we'd like to know just what the whole root system looks like, but we have no way of actually doing that. So we've taken a GWAS approach, and we have a number of candidates. This one's a little further along than most. I won't spend too much time on it. We're provisionally calling it ideal root architecture, but essentially it's a gene involved in central carbon-nitrogen metabolism, and it's broken in this ILP line here. So we've mapped this gene. It's broken in the ILP line. We get reduced expression in brace roots and lateral roots, and any rills that have the ILP allele, the expression is greatly reduced. And so what we think is happening is this gene is involved, again, in carbon-nitrogen metabolism, and we think the ILP lines basically sense that they are nitrogen satiated, and they grow their roots not particularly looking for more nitrogen. So it like fundamentally changes the root architecture because of what they're sensing their status is internally. So we've got a little bit more work to do in this, but the orthologs and Arabidopsis and tomato seem to be doing a very similar thing. So stay tuned for more on this gene. A story that's a little bit further along, but also harkens back to classic maize genetics, is Rootless 1. So a graduate student in the lab, Adam Bray, has been working with this gene for quite a while. First described, it's in Mutants of Maize by Jenkins in 1930. Essentially, it makes no nodal roots, which are the roots that form with each leaf, right? And eventually encompass the majority of the root system. So you can see, especially above ground, there's almost no nodal roots in this plant at all. Adam has identified the gene. It turns out to be a BHLH transcription factor. Not going to talk too much more about that. We have molecular analyses and things like this going on, but more about what it can do to quantitative variation for nodal roots, which is one of the main axes of variation of a root system. And so we've identified a couple of DS insertions, one here at the end and one here, and you can see they have defects in nodal roots, so we're pretty confident this is the right gene. It took us a while to get this. We had to use nanopore sequencing because there's so much structural variation up here, but eventually we were able to identify, classic maize genetic story, several transposons that knocked out the first two exons, and there's even more transposons in this landrace that it came from, which actually in some cases has too many nodal roots. So we've been able to identify that it's actually a problem in nodal root initiation. So you can see here at a similar stage, there are none in the mutants and far fewer here in the coleoptile node. And furthermore, with some in situ work, it seems to be expressed at a similar time as this early marker, WOX5b. So it seems to be involved in specifying the nodal roots themselves. But if you look at a nodal root that's already starting to form, it has a very similar expression pattern to scarecrow here in the endodermal layer. So we think this is a very critical gene that's controlling nodal root development. And so we're next wondering, to what extent can it control quantitative genetic variation for nodal roots, right? That's what we'd like to be able to, the knob we'd like to be able to tune for breeding different root idiotypes for different environments. So we have a lot of this field data, as I mentioned, and we can rank, and you can see there's quite a lot of variation for nodal roots. Going back to the literature, we actually identified two separate genetic QTL studies where RT1 is sitting right underneath some nodal root QTL. One of these is with Teosinte, another is a cross with Gaspé Flint, which is a sort of an unusual landrace variety. So we think these actually are quantitative genetic regulators. I'm cutting out a whole bunch of field work here, but we've done a lot of work with Teosinte populations, and there's some alleles which, as a whole, the Teosinte alleles increase nodal root numbers, although there's some introgressions where they go way up and actually where they go down. And in fact, what we see is a weird developmental sort of over proliferation. Normally the nodal roots form nice and orderly, but in these lines we see these sort of roots that are stuck in between here, out of order. Don't know a lot about how that works yet, but it seems to be tied to simply the expression of RT1 itself. So these are, again, two teosinte alleles that have increased nodal root numbers. We've finally gone back and been able to sequence using nanopore sequencing the promoters. It turns out there's a helitron in one of these overproducers and a cacta element in another. This is brand new data. This is one of these teosinte lines in Gaspé Flint that have too few. So we can see that these transposons seem to be reducing nodal root number. And then just recently we found this hat transposon that have the lowest set, or many of the NAMs that have founders have the lowest set of, or lowest numbers of nodal roots. So we're pretty excited about the possibility to finally have, again, a knob that we can tune for this really important root architectural trait. So I promised you maybe videos or a little more excitement, or at least I insinuated that. So I'm going to give you a little bit more of some of the future cutting edge technology that we're using here. So we've invested heavily in two X-ray image systems. One is an industrial system. It's made for aerospace manufacturing industry. Several of us could stand inside of it, so we can scan really big things in this. Another is an X-ray microscope, which allows us to get down to cell level resolution, and I'll show you some of those videos. So the idea is we can really get a multi-scale data and integrate it. So here's a couple examples of what we can do with an X-ray system. We can literally have X-ray vision. We can through the soil. This is a 3D reconstruction of a corn root system, and you can see we can put a pretty large thing in here. We can also follow organs as they develop, ears and tassels, right? Before they're apparent by eye, before it's popped out of the plant, we can scan these things, and we can scan them over time. At the energies we use, we've never seen any negative effects on the plant itself. Then with the X-ray microscope, we can take, for example, a root crown, and we can zoom in. Here we're looking at the nodal root plexus, where these roots emerge. Here we're looking at a root tip. All these little spots you see are nuclei. So we can literally count the cells in an entire root tip and understand how variation at that level is connected to variation at the architectural level. And similarly with development of these nodal roots. Obviously, the pictures are one thing, but being able to computationally dissect these is another. And so you can see this is, we use a lot of deep learning methods to segment or classify individual parts, tissues or roots themselves, so that we can measure them. We've also done quite a lot of work with floral development. We're interested in vasculature. The resolution on this is even low. The raw data is incredible, but this is a soybean flower. We can see that we can count the pollen grains in the anthers, see the texture on the stigma. It's just a phenomenal data that we're able to get with this microscope, which is something that's really hasn't been used in plant science very much. And again, same idea is that we can start to segment these computationally and then measure them. I'm going to kind of move through these so I don't run out of time, but I don't want to deprive you of just some of this. I mean, I look at this and I'm sort of astounded by it too. Obviously, a soybean seed where we count every cell in every tissue in the seed, and then there's a lot of computationally we can segment everything out. Soteria, inflorescence here. We're very interested in understanding early on if we can identify different branches, so we can segment these branches, and we're starting to try to predict at least which one will go on to be a bristle, which one will go on to be the floral meristeme. Here we're just looking at local curvature. That's what these different colors are, and you can see a lot more curvature in the floral meristeme as new meristems are emerging. That will eventually be the parts of the flower. So there's some really interesting developmental biology, I think, that we're starting to do and that we'll be able to do. But getting back to the roots, information content, thinking about having a pile of 8,000 of these and say, okay, measure the differences. It's not that easy to do. We can get somewhere with 2D, but we're still losing a lot of information. So we've learned that in about 3 minutes we can scan which are stiff and lignified, right? So we've scanned these now by the thousands. We've got a pipeline where we can extract lots and lots of features. In fact, here's are all the features we can extract. We can make this list as long as we want. It's essentially arbitrary, all the things that we can measure about the root system. Here's a study where we've correlated, though, all of these fancy 3D traits with a classic old trait, root pulling. The amount of force it takes to yank a root system out of the ground actually has a lot of correlation with the three-dimensional structure of the root system, which is nice because that's a lot easier to do at field scale. This is a project with John McKay, who literally has a robot to do it. So, you don't even need to do it yourself anymore, apparently. But the upshot of all these data is really that we think of the traits not explicit, trait 1, 2, 3, but really as a root architecture variation per se. And we typically use a principal component analysis to reduce dimensions, and then a linear discriminant analysis. And this allows us a lot of genetic discrimination. So there's 4 samples of types, and you can see they cluster together really well. You can measure something really well. You can often find differences which are a little more subtle than if you have a coarse tool. So we feel pretty confident we can see a lot of variation in these root systems. We've done quite a bit of work. In fact, just published a nice paper with sorghum panicle diversity. It's another shape that's complex. It's very hard to measure by hand. We've done it. But with the X-ray system, we can essentially computationally dissect the whole thing. And so we've done that. We've developed a whole pipeline to measure all the seeds, the shape of the seeds, the 3D structure, the branches, the main rachis. And we looked at this across the major botanical races, trying to understand the phenotypic and genetic relationships between the two. And so what we see actually is really a continuum of inflorescence phenotypes. And by principal component, we can't really tell these apart that well phenotypically. If I hand you a panicle and say, tell me which race, botanical race this is, it's harder to do than we thought it would be. So anyways, more data, but essentially even with fancy phenotyping, we can only get about 70% accuracy classification versus 20% by random chance because there's five races. So not too bad. When we look at all these traits per se, if we look at kinship matrix, you can see they pop out pretty well by SNP data. We see no such patterns when we look at these the three-dimensional morphological diversity. So it's a very unique relationship between the two, which should bode well for future GWAS work that we plan to do. Additionally, of course, we've dug the root systems of these same plants and are just starting to embark on a GWAS study, a co-GWAS study with both panicle and root architecture. So I'm going to run out of time. I won't spend too much more time on that, but these are the different traits we can measure here. We get pretty reasonable heritabilities. So of course, heritability to some extent is a function of how well you can measure the trait. If it's a complex trait, it's plastic, we need better tools to do it. That's the argument I'm trying to make here. So again, any type of inflorescence works pretty well. We've done a lot of work with grapes. Same idea here, measure the berries, measure differences, saw convex holder, very, very useful trait for any 3D shape. You can remove them, measure the rachis. And what we're starting to do now with these data and others is really, as we get high quality, high information content, empirical data, we can start to make predictive models, predictive simulation models. And so we can do pretty well. We can, with the information we can get, we can grow a grape rachis that is pretty similar to the actual rachis itself. So that's definitely where we're headed with that work. Zooming back into the root systems and sort of the hard work, because anything in soil is always hard. I'll tell you a little bit about our efforts to develop segmentation, high throughput segmentation methods. So this obviously is a soybean root system. It's growing in this pot here. We've been able to segment out, so remove the soil, capture the nodules themselves here. So this is just an animation of what that looks like. When you get an X-ray scan, X-rays see everything, right? Nothing's hidden. So we get the soil, we get the different particles, we get the pores, we get any other insects that happen to be in there and the roots. Typically, we just want to capture the roots, although the soil itself is, of course, pretty interesting. Depending on what soil type or substrate we have, it goes from anything from a kind of a hard problem to a really hard problem. Many of these are hard to capture by eye. So, let's turn to computers. So basically, if we give a computer a good labeled training set, it can learn using deep learning methods. This is a UNet method. We can get pretty good automated segmentation. We can go for something that takes 2 weeks for a human to do by hand something in 2 hours, we get an entire connected root system here. And so now we're able to go back and remember the time series approach from the gel, which again has limitations. We're getting to the point where we can now do these same analyses, but in soil. So very excited about that. We can measure, again, individual root traits in a complex root architecture. This is that soybean root architecture. Red is day 15, blue is day 10. But we can also track other stuff like these nodules. I know this is a little hard to see. You can see there's just a few nodules on day 10, quite a few more, and they got bigger. So we can track these individual nodules as well with these methods. And so we've got a pretty good study of about 100 soybean plants at a couple of time points that we've been working on. And this is maybe just, again, you've seen these type of data before, but we can zoom in with the X-ray microscope and measure individual cells in the bacterioids and segment these out. We'd like to capture some functional information as well, not just structural. And so I showed you the PET data earlier. That's sort of large scale data, but on a subcell or cellular resolution, we've been working with a colleague, David Fike, who has a SIMS, it's a mass spec imaging method, and you can the mass spec for any element you want. You can basically any chemical compound you want, any structure. Here we're just looking at nitrogen. And so you can see the intracellular nitrogen pattern within the bacterial cells, and these are the bacterial cells, and these are the plant cells here. So we haven't gotten too far here, but again, the idea is to pair this functional and structural imaging. And ultimately, we'd like to be able to understand resource exchange dynamics between bacteria and plants within a nodule, or in this case, arbuscular mycorrhizal fungi, which can be considered extensions of the root system. And so here you can see spores, hyphae, and those really dark areas in the root are hyphae that have invaded the root system. And so we're able to actually quantify these networks as they're invading the root system, pair them with functional methods like PET, like SIMS, and understand resource exchange dynamics at a microscopic level. So here's just the whole structure where we've segmented out. Yellow is the fungus, gray is the root system there. Okay, so I've got a few more minutes. So I'd like to spend a little bit more time on PET, which I think has an incredible potential, especially paired with CT. This is a very early study I did. This is an imaging artifact here, just because we had kind of not a high-tech PET imaging system. But we essentially feed this corn plant radioisotope, carbon-11, CO2. It has a 20-minute half-life, and as it decays, it makes some energetic particles that can be detected on the PET ring. So we can literally, it's sort of a pulse chase experiment. We give it 5 minutes of radioactive gas, we flush it out, and we say, where does that 5 minutes of synthesis, where's it going? Where is the plant deciding to put it? And you can see it puts it down into the root systems pretty quickly. Here's what the inside of the machine looks like. So again, this is a fantastic interdisciplinary group here, and you guys are you're definitely on the right track. It's hard work to learn to speak the languages of, in this case, nuclear physicist, who's my collaborator here, Jan Chantai, or engineers or computer scientists. But if you do, you can really open up new avenues in plant science. So he had the foresight to build one of these fancy detectors. You can see inside of a convirant growth chamber, we can do these plant studies. And so this allows us now to capture carbon dynamics in real time. We get about 2.5 hours here. These are 3 corn plants, or subjects as they call them at the med school. But because it has such a short half, it's gone by the next day. And so we can actually track over developmental time as well. We can do the same experiment day after day after day. And so we can see, for example, no carbon is going to the root system early on in development, presumably because the sugar is coming from the seed. But when that runs out, we see it moving down into the roots here. So again, it just gives us some insight that would be hard to capture otherwise. And paired with CT, this is again that soybean root system. It's kind of a low res, but here are the nodules here. So we can actually measure how much carbon is going to a nodule versus going to, say, a root tip to drive root growth. So when a plant enters these relationships, how much is it giving up, right? And how much is it getting back? So we can use a nitrogen isotope to measure how much nitrogen the plant is getting itself. And we can do this in real time and dynamically and structurally. We've done some similar work in Metacago. This is infected by AMF fungi, and I think it's kind of hard to see here, but the idea is over time we can see this hotspot here, region of interest. This is a prime sample that we would cut out and put into the X-ray microscope to really understand why there is so much carbon going there, presumably because of extensive AMF colonization. Okay, I know I've taken you guys on a whirlwind tour, but one more little piece, zoom back out actually. It's always been incredibly frustrating to me that we can't actually know the three-dimensional structure of a freely grown root system in the soil. There's really no way to do it. You can dig a trench and dig it up piece by piece. You don't really get that three-dimensional aspect. Furthermore, it'd be great if we could put a bunch of sensors in the soil and understand these structural functional relationships. So we basically just went to we basically designed a system where we could do this. Pretty low tech, but we're getting sort of better and better at it. So we started with a big sack full of turfus, right? So we're not using soil yet because turfus washes off the roots pretty easily. And then we built a 3D grid, so basically fishing line and anything. And this is a sorghum root system. You can see the entire, once we cut the bag open and remove the soil, we're capturing the entire root system here. We've gotten a little bit more sophisticated with, so we have a greenhouse full of these mesocosms, is just a generic term for sort of a mid-scale system. We've designed these things. We can, of course, make them any size, height, or width. We now have this nice PVC scaffold inside. We have drainage holes. We can collect runoff. And then we just put as many sensors as we can in them. So if you have a sensor and you want to test it out, this is a really good way to do it. And so we found if we put sensors sort of in this configuration here, so we have matrix water potential sensors, also measure temperature, a Picaro gas analyzer to measure root respiration or microbial CO2 production if we decided to put microbes in here. And again, we get this dynamically. So here's temperature, soil water potential, and sort of a four-dimensional map. These are two switchgrass genotypes, WBC and VS16. And you can see that they're interacting a little differently. They're pulling water out a little differently. So we can't see the roots at this point, but at the end of the experiment, we basically open the side of the container, we get a shop vac and maybe some brushes and stuff, and we just slowly remove the turfus in this case. It'll be harder with soil, but we'll get there. And so we can see if we grow this variety at -1.5 megapascals, which we can control with these matrix water potential sensors, we know exactly that we are holding it at this. We just see a profound difference in the plant's decisions on how it's growing. The roots get much, much wider. They're much, much deeper than this. I know you can't see it all, but clearly there's big gaps here. So the root system has decided to go really exploratory rather than filling in a dense area, which makes sense. But it's just that we can actually see it and quantify it here. Here's those two genotypes. Hopefully you can see. It's awkward to take pictures in this thing, but you can see that there's two very different root architectures here. So how do we turn this into data? I'll tell you in a minute, but here are three-dimensional models, and these are things that all those tools I talked about before with the root crowns, et cetera, we can use those exact tools, but now we can do it on literally the entire root system, which has been growing largely uninhibited in this box. So now we can really kind of know what's actually happening, right? The way we get these 3D models is through photogrammetry, or commonly referred to as structure from motion now. It is basically Tyler Dow, the postdoc in my lab, with a digital camera taking thousands of pictures. And we can use a GPU basically to model the camera geometry and ultimately to model the root system itself. You see all those crazy patterns and stuff. In fact, he got his son with a can of paint to just slap paint all over like Jackson Pollock style. That's because we need unique models for each image. If there's unique patterns, that allows the GPU to put the pictures together much more efficiently. And then again, we have to segment out the room, this crazy room, so we can, and the scaffold itself, so we can see the root systems. Ultimately, we want to put these back, or we started to do this, but just to put these back into the environmental models that we've made from the sensor data, and really understand. Again, we'd expect areas that were sort of getting dry to have a large root mass, for example. That would be a simple hypothesis. And the last little bit of time, I just want to tell you how we're ground truthing these things. It's pretty straightforward. We have a grid. We can basically just go piece by piece all the way up and cut the pieces out, measure the biomass or other root properties in that grid, and get a map of it. Ultimately, we're leaving this root crown here because, you know, one question is when we're in the field and we dig up the root crown, how good of a representation is that of the whole root system? And so we will scan this root crown by X-ray, measure properties of this root crown, and ask, can we predict any properties of the actual root system? And we can do this because we have the actual root system. So that's the ground truthing. We get kind of a 3D model of the ground truthing. So we can do this for these root crowns, but we can also do it for the other tools, soil coring or mini rhizotrons, right? We can, in silico, take 1,000 soil cores or mini rhizotrons and ask, does the data from these mini rhizotrons tell us anything about the actual root system? And I think that's going to be a really powerful approach to helping us understand the biology. So I think I'm about over time. So yeah, this is the lab. It's a big venture. So again, my model's a little different, which is bring everyone into the lab. So I have physiologists, quantitative geneticists, molecular biologists, applied mathematicians, computer scientists, engineer types. And so it's a big group, and we've learned to work together. And just a shameless plug, if you're interested in roots, next spring, come to Columbia, Missouri, where we're hosting for the first time in the US, the International Society of Root Research, which is a symposium, which is an incredible symposium. And so stay tuned for that. You can look it up on the web to see some the speaker list as it develops. So I don't know if I have time for questions or not. If so, okay. I'll be here all day. Happy to talk to you. Final parting shot. Some of this stuff, because we so much visual data, we've developed an extensive array of visual reality tools. I think with coronavirus going around, now is not the right time to do this, to put the headsets on, but we routinely do this, and I'll bring it the next time when it's a little safer. This is a little virtual farm where we even started to embed our data. So you can go to the farm, you can, like you always wanted to do, pull up a soybean plant, but the roots are actually data. They're X-ray scans from our real data. We're putting these two things together. So it's quite an experience. Okay, so I'll stop there. Thanks very much for your attention. And yeah.
Wow, that is pretty awesome. Let's give Dr. Topp a round of applause. I think we'll give time for one question if anyone has a burning question. Heart's desire.
So I'm actually going to do a two-part question. Thank you for the talk. Those images and videos were incredible. The first question is, have you guys started scanning collections of germplasm to submit so that you can get more eyes, more researchers looking at it? And then my second part is, what's the computational time? So in the very end with the grid system,
yes.
Like what processing time for something like that?
Yes, I'll answer the second one first because it's pretty straightforward. I mean, of course, it depends on the size of the computer, but let's say on a $1,000 gaming computer, which is a serious computer, it might take overnight. If we put it on our deep learning computer, it goes a lot faster. So it just depends, but it's a reasonable amount of time. We get about 12 of these, but it's every 3 or 4 months, right? So it's not like we're generating mesocosm data every day. It's a lot of work to move things in and out. So, but it's a reasonable amount of time. Back to your first question, I'm extremely keen to scan as much germplasm as possible. So we've done some work where we've talked about doing wheat panicles. One of the most interesting things we've done is scan some maize artifacts from the Anthropology Museum in Mexico. And so we scanned a 1,500-year-old and a 5,300-year-old maize root crown from a cave in central Mexico that was preserved. And yeah, so that's the type of thing that I think X-rays are really, really good at. They can preserve this variation. If you have ideas for stuff, I would love to talk to you about it. Okay, thanks again.
That's awesome. Dr. Topp, we really appreciate you coming up here for our symposium, and all of the work that you've done is highly valuable. I mean, I love digging into your research. So, okay.